Part 3. So, there's this idea of a thing called the RNA world, and it's probably not as simple as that. But at the same time, I wasn't around 4 billion years ago or 5 billion years ago, so I don't know the answer to how everything got the way it is. Yeah, don't blame me. But there are people doing experiments to try to figure this stuff out, or at least see how things can evolve. And so a lot of these experiments use viruses because they're so simple. So one of these viruses that they use is a virus called Q beta. So that's Q and this sort of squiggly B on a stick. And Q beta is a tiny bacteriophage, and it has a little bitty genome, and it encodes a polymerase protein. That's the shape of a polymerase, and it encodes a capsid protein. And that's about all you need. If you're Q beta, you can go through your life. Polymerase copies your genome, capsid packages your genome, and you can make more polymerase when you get to the next cell. What they did here, though, was they did an experiment where they took the protein, the polymerase protein, so that's the little blue pentagons, and they took the virus genome, and that's the red squiggles, and they put them into solutions that had free nucleotides that could be joined together to make new copies. And they let those go. And sure enough, with these little sort of primordial soups, the virus components were able to copy themselves. Polymerase would copy RNA, make more polymerase, copy more RNA. I think they had translation in there as well. And you could actually passage this. So they passaged it over and over again, which means they took a little bit out of this one and put it into a new vat of nucleic acids and all the stuff of life. And what they saw over time is that instead of the virus just copying itself and making nice full-length copies, they got this thing, which they called Spiegelman's monster. Named after the third author on that paper. It's Mills et al. 67, and you can read it, but it's from 67, and they didn't have the sort of graphics that we have now for explaining things. Yeah, that sort of stuff was harder. All the details there, though, and there are lots of nice commentaries about it, like the one that I stole this from. Anyway, what they found was that instead of getting the full-length virus back out, they had some polymerases, and most of the virus had been replaced by a parasitic version of itself. So instead of the complete virus, so shown here is a complete plus-stranded Q beta genome, you ended up with a parasitic version of that had both ends of the genome and no middle of it. What's the advantage of this thing? Well, if it takes a particular amount of time to copy each base, then a shorter genome can be copied faster than a larger genome. And so, whenever a mistake is made that shortens the genome, some sort of recombination, or, there's not really a good deletion mechanism, so it's probably recombination. Then you get a little shorter, and each time the shorter one will outcompete the longer one. Isn't that weird? Now, you can actually do this experiment two ways. So Spiegelman's monster was the name that he gave to this particular thing. I think it was around 200 nucleotides long, that just came to predominate in his little primordial soups. It was a parasitized version of a virus, which is a parasite itself. And there was another experiment that was done a year or two later, published in the same journal, PNAS, where they took nucleotides and bits of tRNA and stuff like that, and polymerases, and they assembled them from raw material, and they kept feeding it back more raw material and took the assembled things and transferred them. And they were able to come up with something with the same sort of properties. It was a short, non-coding RNA, something like a viroid, which was able to be copied, but which didn't actually convey any other information. And so it was essentially a parasitic RNA. So if you start from genes, if you have some RNA around and some proteins around, this is both kind of the ultimate goal of viruses, maybe to replicate faster, but also this is a thing that will directly compete with viruses for replication. Like, they're sloppy because that's how they are, and there are some advantages to that, but sloppiness comes at a price in that you make parasites that will eventually destroy you. So how is it that viruses aren't completely extinct? You're probably asking yourself, like, yeah, what? So it turns out that most viruses, particularly small RNA viruses, which are most susceptible to this kind of recombination and shortening and just mayhem, they tend to replicate in small compartments. A lot of them will carry proteins to make a small compartment in the cell in which to replicate. Now, there was an experiment. It's a paper that's cited in the paper that's cited on the previous page, but they actually took this same virus RNA, so Q beta RNA, and so they made the replication protein and they gave it all the necessary things to copy itself. And they found that the replication protein and genome pair could go on replicating and making copies of themselves. Make RNA makes protein, makes RNA makes protein for a little while. But after an hour, you would end up with these little Spiegelman monster type things, little shortened versions of the RNA that were parasitic, and they would crowd out the main replicating lineage, and replication would stop at that point because you'd have so many little parasites that couldn't make a polymerase that the few polymerases that were made mostly ended up making copies of parasites. And so you didn't get any new copies of the RNA that could make more protein to make more of the viruses. And so these things just drive themselves to extinction. But you're saying to yourself, but yeah, viruses, I wish viruses would drive themselves to extinction, right? But they haven't yet. And I don't think they're going to. So most viruses will actually create small chambers. And so in this experiment, what they did was they created small chambers. And they found that, yeah, occasionally these little monsters appear, mini monsters, mini genomes, parasitic genomes, whatever you want to call them. But if they're contained inside of a smaller thing, then they only poison one little part. And since it's rare that these things will appear, and very common that they multiply once they appear, as long as you keep them separated, most of these others, where the mini monsters don't appear, can replicate themselves, and they're okay. It's just when everybody's all together and can mix around, that's when you get into trouble. So, big takeaways from this, and these are worth thinking about, and these are going to be worth talking about. We're going to talk about these in class a little, because they take a little while to get your head around, I think. So, viruses live in an error-filled world. And error and recombination can accelerate evolution, and that can be good if the virus is facing an evolving challenge, like an immune system. So viruses of humans, for example, we have great immune systems that are constantly adapting to try to pound these viruses into the dirt. And yet, the viruses survive because they make mistakes. They're sloppy enough that they can make enough mistakes to avoid the immune system for just long enough to be able to grow and then be spread to another person. So that is a lifestyle choice, and it's a dangerous one. It comes with risks. We can also see from this, and it's actually true, that viruses depend on signals. Usually these signals are going to be outside of the genes, like in between the genes or at the beginning or end of the genome. That's going to be how they recognize their RNA, because the virus copies itself. It doesn't copy any random RNA in the cell. It's going to have special signals that it binds to. And so that's actually true, yeah. But whenever the virus does a recombination that shortens the genome and it loses some of that coding sequence, it actually makes a smaller version that can be replicated faster. And these parasitic nucleic acids are actually very well known from every kind of RNA virus that's out there. And they arise spontaneously, usually by combining they keep the signals that you need for replication and lose the middle. And it's not that they're like smart and they've figured out that, oh, these are the parts I need to keep. It's that the virus makes random mistakes. And when it makes a mistake that destroys one of the signals it's looking for to recognize itself, then you just have a dead virus and yeah, nothing happens. When it makes a mistake that makes a shorter version of the virus with all the signals intact, these are our little signals here, then that thing can be copied. And since it's shorter, it can be copied faster than its parent genome or host genome. And so that becomes a parasitic RNA form, like a parasitic genome or mini genome. So, yeah, the price of this accelerated evolution is that you will start to make miniature versions of the virus that will then compete with the virus and eventually drive that virus to extinction if it stays still. So there are two things that viruses do that are probably strategies in the sense that nobody's thinking of these things, but they have this effect. So we can think of them as strategies. Like if people were doing this, yeah, it would be a strategy. One of those is compartmentalization. So just do your replication in a small chamber. And if something, have lots of those small chambers, and if something bad happens in there, that's fine. You know, never let anything out. You have some kind of quality control, and you just shut that down. And most RNA viruses do this. The other alternative is that, you know what? You can let this kind of situation happen. And you can just package all these things up. And you know what? If you package every one of those up into a little virion and send them to a new cell, there are so many other cells you could go to that you're not going to end up in the same cell as one of these little mini genomes, probably. And if a mini genome lands in a cell, it doesn't encode any proteins. And so nothing's going to happen. It's just a little chain of RNA, and RNA doesn't last long in this world without something to replicate it. So they all go out and die, whereas actual genomes that encode things can go out and do the whole cycle again. So you could look at the virion as a mechanism for purifying the genetic lineage and driving out all these little mini genome parasites that happen as a result of having an error-prone replication style. Isn't that weird? Maybe look over this again, maybe listen through this again, maybe think through this again. Draw it out on a piece of paper, whatever makes you, get it. But this is a big concept in virology, that viruses are parasitic but spawn their own parasites and require strategies to keep those subviral parasites at bay, lest the viruses be driven to extinction themselves. So, it's viruses all the way down, basically. Viruses of organisms, viruses of viruses, satellite elements of viruses, and parasites of satellite organisms of viruses of hosts. Something like that, yeah. Once you get that, and once that makes sense, you understand a fundamental idea about how viruses work. These are not smart beings. They are probably the dumbest bits of biology you will ever run into. They work through random mutation and through chance and error and making lots and lots and lots of copies. They are fast and sloppy in everything that they do. And once you get that, you will have a much better understanding and an ability to kind of get inside the empty little heads of these viruses. Yeah. So I hope that helps. And thanks very much for watching.